So it's great to be here. I wish I was there in person, but it's good to join virtually. So I'm going to be discussing GeoSeq, which is a platform that we've developed for global pandemic early warning systems and for pathogen and drug resistance surveillance. We developed this platform together with the Rockefeller Foundation and in affiliation with Metasub. And this is powered by Biotia, but we are also launching GeoSeq Foundation currently. So this is a nonprofit public application. So the problem that we are addressing with this platform is that 14 million people die annually from infectious diseases. We believe, as many of you do, that infectious disease is best controlled by prevention, not by cures, but we cannot prevent threats we are blind to. We see that next-gen sequencing and genomics is revolutionizing rapid diagnostics and drug resistance profiling. Unfortunately, many pathogen monitoring programs that exist are often inequitable, limited, and siloed. And then we also see that climate change is driving pathogenic risk, especially in low and middle income countries. In terms of climate and health, we see changes in climate are driving infectious disease risk, especially for vector and waterborne pathogens. And climate change is also impacting antimicrobial resistance. And so this is happening in a number of different ways, including changing the distribution of vectors, malnutrition of hosts, natural disasters impacting sanitation, and stress on livestock and overuse of antibiotics. So I'm going to talk a little bit about the platform that we've been developing, as well as some pilots that we've been running. So this is a video showing the GeoSeq platform. On this platform, we connect varied data, including microbial genomics data, climate and epidemiological data. And we use these varied data streams to build predictive models. So we build these geospatial models to map and track infectious diseases, and then also model the impacts of infectious disease, of climate change on infectious disease. So I'm showing here, for example, different climate variables such as temperature and precipitation. The platform has more than 200,000 metagenomic samples on it currently. And as you can see, there's a search going on here. So you can query by species. You can look for different metadata such as location, and also search by DNA sequence. So right now, this is showing a search of the MEK gene in different locations. But you can also search for like an emerging genetic marker that you're interested in to see if other researchers globally are finding a distribution of that marker. The platform's been built out for researchers, and then we also have tools that have been developed for public health agencies. So on the research side, researchers can connect, query, visualize, and analyze data as being shown here. So these are some basic tools for example, K-mer distances and taxonomic abundances. There's also a section for apps for deeper analysis. And for folks that are on the research side, Ford Combs, who works with us at GeoSeq, will be giving a workshop tomorrow morning for researchers. So really digging into how this can be useful and how to use it. On the public health side, we've developed different analytic tools and dashboards. So this is more traditional epidemiological statistics, such as dengue case count or infectious disease case counts. And then we also look at covariates such as GDP. And so this is in open beta, and the platform's available for use. So I'm going to highlight some different projects that we've been working on. First, I know that a number of people here have potentially been involved with the Metasub consortium. We are also involved as researchers with this consortium. Metasub is the international consortium mapping microbes across the world, often in urban environments, coming out of Chris Mason's lab at Weill Cornell. And so there's a number of publications coming out of this work, but just to highlight one here, there's the global metagenomic map of urban microbiomes and antimicrobial resistance. published in Cell in 2021. And a lot of the work that was done on that platform and the analytic tools have been migrated and evolved into GeoSeq. So you can see for example, we have samples in Seoul that are on the platform that are available for use and analysis. Some other projects that we've been doing at GeoSeq Foundation is we have been funding different new watchtower sites shown here on the left. So these are new sites that are generating data, often in data deserts or high-risk areas. Some of these we've been working with to sequence patient samples and wastewater samples with the goal, at least for one project in the US, is mapping AMR distribution. I'm going to talk about this a little bit more. We have some hospital partners that were using these data to impact antimicrobial selection. Another project that we are just starting to launch that we're excited about is working with Institut Pasteur in Cambodia on environmental surveillance in wet markets and abattoirs. And this is to identify zoonotic pathogens earlier with the impact of flagging novel pathogens, identifying transmission patterns, et cetera. And then some work we've been doing together with our partners at Fiocruz in Brazil is sequencing mosquitoes for arboviruses. And so for this work, the outcome is finding areas with high outbreak risk and using those data to drive data-driven mosquito control programs. So where and when to spray for mosquitoes. For that work in dengue, we have been modeling the distribution of Aedes mosquitoes because we are mostly focused on dengue with this work. Some of the work is also looking at malaria and other mosquito-borne viruses. For the predictive models, because there is this very clear link between dengue and climate, we have been modeling distribution of these Aedes mosquitoes. We found that when we move from a climate-only model to a multimodal model, we get greater precision, as shown here. So as we moving from, here, actually, let me go back. As we add in land use and sociological data, this really improves the resolution of our spatial maxent mosquito model. And we're working to extend this model to make counterfactual predictions of dengue case counts. And we're doing this in different ways. So we're building generalized lasso linear models and looking at other causative factors as well. So again, this is to inform local data-driven spraying of mosquitoes. And then in terms of the drug resistance mapping that we're doing in the US, we have a couple hospital partners that we've started working with, and we're working with their antibiotic stewardship departments to provide data for selection of antimicrobials. So for example, we have a hospital in Connecticut that we're starting to work with that have a research program going on, that they have an app that they've developed for clinicians. They provide guidance on selection of antimicrobials for these clinicians to decrease the drug resistance rates that they're seeing in the hospital. Right now, they're actually using national level data on selection of antimicrobials. So we're really trying to bring them from the national level, at least down to the regional level as a first step. But the goal is to move to the hospital level or patient level, right? So we want to see precision medicine for infectious disease. We have a number of users that we're supporting across the world. So we have the Watchtower programs, which I mentioned are newly funded sites coming out of GeoSeq. And then we are partnering with Pasteur Network and providing support for there are 33 institutes across the world. And then the MetaSub Consortium, which is mostly focused in urban environments, is another group. And we're providing wet lab protocol standards, dry lab tutorials, as well as bioinformatic expertise. And in some cases, really providing like a sample to result sort of support, or in other cases, a lighter touch sort of support. And I'm gonna leave it there. I just want to thank Sujin for the work that we've done together and for inviting us to present, and the folks at Biotia, Rockefeller Foundation, Weill Cornell, MetaSub, and our other partners as well.